Let's skip the fluff and violently accelerate right into the high-yield chemistry you need to guarantee full marks on paper 2. Lock in, get your screens ready because this is the ultimate exam sprint explainer. Right, Section 1, Rates and Reversible Reactions. Let's go.
Examiners absolutely love testing mean rate, and you've got to remember the core drivers of reaction speed. Increasing surface area, like crushing solids into powders, bumping up the concentration or pressure, or cranking the temperature, they all do one vital thing. They increase the successful collision frequency. Oh, and for your graphs, remember to draw a tangent on that curve to find the exact rate. Just divide the change in quantity on the y-axis by the time on the x-axis. Easy marks.
Now, you've probably done the classic disappearing cross experiment. It brilliantly illustrates how hotter temperatures don't just speed up particles so they collide more often, but critically, it gives them more activation energy. As the reaction makes a cloudy precipitate, turbidity shoots up until boom, that cross disappears. Less time on the clock, that means a faster rate.
Okay, moving to reversible reactions. Le Chatelier's principle dictates that if you change the conditions, the system is going to adjust to counteract that change. It's like a chemical tug-of-war. The crucial point here is that increasing the temperature will always favor the endothermic direction because the system is desperately trying to absorb that extra heat. Conversely, dropping the temperature, that's going to favor the exothermic, heat-producing direction.
Section 2 already, Organic Chemistry and Polymers.
You have to know the difference between alkanes and alkenes. Alkanes are saturated chains. They've only got single bonds. Alkenes, on the other hand, have a carbon-carbon double bond, which makes them unsaturated. Now, the ultimate exam hack here is the bromine water test. Only an unsaturated alkene will react and turn that orangey-brown liquid completely colorless. Alkanes, they're boring. They just stay orange.
Think about it. Crude oil is a totally useless mixture until we separate it out using fractional distillation. This sorts the hydrocarbons by their boiling points. The longest alkanes have the absolute strongest intermolecular forces, meaning they condense right at the hot bottom of the column. But here's the thing, we have a massive demand for shorter, highly flammable fuels, so we literally crack those long, viscous alkanes into smaller, useful alkanes and alkenes. You can do this with catalytic cracking at 550 degrees Celsius using a zeolite catalyst, or use steam cracking at over 800 degrees.
Triple science alert. If you're taking the triple paper, listen up. Adding an OH group to a hydrocarbon makes it an alcohol, like ethanol. But if you oxidize that alcohol, without combustion, mind you, it creates a carboxylic acid. That shifts the functional group to -COOH, giving you something like ethanoic acid. You know, the stuff that makes vinegar smell like vinegar.
Another triple alert here for advanced polymers. You need to remember that condensation polymerization literally chucks out a water molecule when joining different functional groups together. So reacting an alcohol and a carboxylic acid kicks out water to make an ester. And don't forget your naturally occurring polymers. Amino acids contain both amino and carboxyl groups to form proteins. And DNA, it's just a complex natural polymer made up of 4 nucleotide monomers twisting into a double helix.
Moving on to Section 3, Chemical Analysis and Testing. Pure mark scheme memorization here.
Chromatography separates substances in a mixture. The mobile phase, usually water, rises up the stationary phase, dragging lighter particles further up the paper. But crucially, you must draw that starting line in pencil. If you use a pen, the ink is going to run right up with your mobile phase and ruin the whole shebang. To find the retention factor, or RF value, just divide the distance your spot moved by the distance the mobile phase moved. Simple as that.
Now, how do you perfectly identify the 4 core gases? You are 100% going to see these on your paper, so lock these exact phrases into your brain right now.
Seriously, say them out loud with me right now. A burning splint causes a squeaky pop for hydrogen. A glowing splint will relight for oxygen. Carbon dioxide turns lime water cloudy when you bubble it through. And chlorine bleaches damp blue litmus paper totally white. Pop, relight, cloudy, bleach. Do not mix those up.
Take a mental screenshot of this table right now. Knowing your positive metal ions is basically non-negotiable for a top grade. For flame tests, you've got crimson lithium, yellow sodium, lilac potassium, orange-red calcium, and green copper. Then for your sodium hydroxide precipitate tests, copper 2 makes a blue precipitate, iron 2 is green, and iron 3 is brown. Notice that calcium, magnesium, and aluminum all make a white precipitate, but here is the trick. Aluminum is the only one that will dissolve if you add excess sodium hydroxide.
Look at the pattern for your negative halide ions when you test them with silver nitrate and nitric acid. They go from white to cream to yellow as you move down the group from chloride to bromide to iodide, white, cream, yellow. And for sulfates, mixing them with barium chloride and hydrochloric acid produces a really distinct white precipitate.
Triple science alert. Let's talk flame emission spectroscopy. It's basically flame testing on steroids. It's an instrumental method, which means it uses expensive lab equipment to get hyper-accurate, incredibly sensitive readings super fast. It passes the light from a flame through a spectroscope to identify the exact wavelengths emitted by specific metal ions. No more guessing what shade of red you're looking at.
All right, Section 4, Earth's atmosphere and resources. Let's look at the big picture.
Our early Earth transitioned from a totally hostile volcanic greenhouse into the beautiful oxygen-rich environment that keeps us alive today. Billions of years ago, volcanoes pumped out nitrogen, CO2, and water vapor. As the Earth cooled down, that water vapor condensed to form our oceans. Huge amounts of CO2 dissolved into those oceans and eventually got trapped in sedimentary rocks. Finally, algae and plants evolved, converting the remaining CO2 into oxygen via photosynthesis.
Now onto our water supply. Remember, potable just means safe to drink. It doesn't mean it's chemically pure. To make fresh water potable, you pass it through filter beds to strip out the insolubles. Then you sterilize it using chlorine, ozone, or UV light to kill off any nasty microbes. If you're in a country without fresh water, you're stuck using seawater. That means you're going to need huge amounts of energy to run desalination, either through distillation or reverse osmosis.
Don't get your wastewater treatments mixed up. The heavy solid sludge that sinks to the bottom requires anaerobic digestion. Meanwhile, the lighter liquid effluent sitting on top, that needs aerobic treatment before it's safe to release back into the environment. Also, keep your atmospheric pollutants straight. Carbon monoxide is odorless, colorless, and fatally bonds to your red blood cells. Sulfur dioxide causes acid rain, and nitrogen oxides cause terrible breathing problems.
Last stretch, Section 5, Materials and the Haber process. Let's crush this.
When we look at polymers, the difference all comes down to the bonds. Thermosoftening polymers melt when you heat them up because they only have weak intermolecular forces between their chains. But if a polymer refuses to melt, even in a blazing hot furnace, it's thermosetting. And that's all thanks to those unbreakable crosslinks permanently locking the polymer chains together.
Final triple science alert, we're talking about the Haber process. This is how we make ammonia for fertilizers. You take nitrogen straight from the air and hydrogen from methane or electrolysis. Then, you pass them over an iron catalyst at a pressure of exactly 200 atmospheres and a temperature of 450 degrees Celsius.
So why is 450 degrees considered a compromise? Well, a lower temperature actually gives a higher yield, but the rate is just way too slow. 450 perfectly balances a decent yield with a fast, profitable rate to churn out those NPK fertilizers that quite literally feed the world. Lock in these facts for paper 2, and you'll crush it. But before we wrap up this explainer, I want to leave you with this. As our global population absolutely explodes, how will industrial chemistry have to evolve to feed billions more, without utterly destroying the atmosphere we just learned about? Good luck on the exam, you've got this.